Good day from Chem Help ASAP. Let's discuss how the safety risks of oncology drugs are handled somewhat differently from other drugs. Upon completion of this video, students will be able to contrast safety concerns of oncology drugs with other indications and describe the design of early trials for oncology drugs. Most approved drugs are designed for indications that do not pose an immediate threat to a patient's life. These drugs still address a medical need. Since the drug may not be addressing an immediate severe health condition and may be taken for prolonged periods of time, drugs are expected by patients and healthcare staff to show low toxicity. Drugs for oncology conditions, cancer drugs, are different. First, cancer does indeed pose a threat to a patient's life. Depending on the cancer, the threat may be immediate. Cancer is a broad disease condition in which a patient's own cells begin dividing uncontrollably. Cancer can therefore be extremely difficult to treat, as cancer drugs must show selectivity for affecting tumor cells, which are the patient's own cells, without overly affecting the patient's healthy cells. That is a difficult selectivity challenge and a reason why oncology drugs often show high toxicity. Because of toxicity observed in virtually all cancer drugs, clinical trials for oncology drugs are designed differently. So here is a table with some different criteria for trial design on the left, and two columns to contrast typical clinical trials and then oncology trials. Let's first look at the test subjects. In a typical drug program, the subjects are healthy volunteers. In contrast, oncology trials initiate with cancer patients. Furthermore, the patients have often received treatment with other cancer drugs. This is a key difference and drives many of the other differences that we see in the trials. In terms of the starting dose, phase 1 trials start with an MRSD determined from the NO-AEL of preclinical toxicology studies, with the MRSD expected to show no toxicity. Efficacy will also likely be negligible. For oncology drugs, the subjects are patients. It would be unethical to give such a low dose that the patients would not likely have some benefit. The starting dose in oncology trials is therefore higher than the MRSD calculation would allow, and therefore the starting dose will also likely show some toxicity. Because of the high likelihood of toxicity, it would be unethical to study oncology drugs in healthy volunteers. Finally, the dosage for patients in initial trials is limited by an exposure cap based on safety observed in preclinical species. In oncology trials, dosing can be increased to determine the level of toxicity that can be tolerated by patients, the so-called dose-limiting toxicity. Such extreme measures for pushing doses are warranted only in severe conditions like cancer. One final comment. The line between phase 1 and phase 2 trials is less clear for oncology drugs because even the earliest clinical trials for oncology drugs aim to achieve efficacious dosing levels. We have contrasted how oncology drugs, because of the challenge of achieving selectivity, generally have greater toxicity than drugs for other indications. We have also described how this inherent toxicity affects the design of clinical trials for oncology drugs. As always, I appreciate your attention, time, and watching all the way to the end of the video. I hope you enjoyed it. Take care.